Hello students, welcome back. We are dealing with the chapter 10 Microbes in Human Welfare, and we have come to the last part. This is the last lecture on this topic, that is lecture number 8. So previously in the last video we have discussed the use of microbes as biocontrol agents. Okay, so coming to the last one, that is use of microbes as biofertilizers. So by now we are aware that the overuse of chemical fertilizers to meet the ever increasing demand of agricultural produce, it has resulted in pollution of soil and water also, and moreover they are also more expensive, right? So there is a large pressure to switch to organic farming, that is nothing but to use these biofertilizers. So let me tell you, what do you mean by the term biofertilizer? So biofertilizers are organisms that can enrich the nutrient quality of soil. So bio means living organisms, right? So here we use certain organisms, living organisms, to increase the fertility or the quality of soil. So the main sources of these biofertilizers are, they include the bacteria, right, and the cyanobacteria. Also certain fungi are also used. So let us study the use of these different biofertilizers one by one. So first one, coming to bacteria. So bacteria are generally used as nitrogen fixers. So there are different kinds of bacteria, and these bacteria are also called as the nitrogen fixing bacteria. Okay, so the nitrogen fixing bacteria, sometimes they may be free living or also they are symbiotic in nature. So first let us study what are these symbiotic nitrogen fixing bacteria. So one good example I can give you here is rhizobium. So it is one of the most important source of biofertilizer, that is using rhizobium. They form an efficient symbiotic association with the leguminous plants. The legume plants, previously and all you have studied about the root nodules which are formed in the leguminous plants, which help in nitrogen fixation, right? And you also in detail you have studied how the root nodule formation takes place. So these nitrogen fixing bacteria, they fix up to 100 to 150 kg of nitrogen per hectare of land. Okay, so the fixed nitrogen here, it is in the form of nitrates and it is used up by the leguminous plants. So it is nothing but these rhizobium, as I told you, where they are living children, they are present in the root nodules of these leguminous plants. Okay, so here in these root nodules, these rhizobium, they fix the nitrogen in the presence of the pink pigment, that is leg hemoglobin, and in the presence of the enzyme nitrogenase. So in detail you have studied it, right? So it is believed that the rhizobium has a nitrogen fixing gene in it. It is also called as the NIF gene, NIF gene. And the pink pigment, the leg hemoglobin, it acts as an oxygen scavenger, and it keeps the anaerobic condition inside the nodules, which activate the enzyme nitrogenase. Okay, so this helps in reducing the nitrogen to ammonia. Okay, so that is why we can call it to be an important source of biofertilizer. So this is about the symbiotic nitrogen fixing bacteria. Next, the loose association of nitrogen fixing bacteria we'll see. So there are certain soil bacteria, they are present living in close contact with the roots of cereals and grasses. Okay, so they also help in nitrogen fixation. So this association here, it is loose mutualism. You can also call it as an associative symbiosis. They are not completely attached. As I told you, they are in just a close association. Okay, so example here you have azospirillum. So the bacteria here, they may reside in the transition zone, that is between the soil and the root. So and sometimes they also enter the roots. So some of the fixed nitrogen here, it is absorbed by the roots, and in turn the bacteria get carbohydrate from the roots. Okay, so the bacteria provide nitrogen and they absorb the carbohydrate from the plants. So this is how both are benefited here. Okay, so example, as I told you, azospirillum is an example here, that is for the closely associated nitrogen fixing bacteria. So these are present in association of roots in certain grasses and maize. Okay, then you have Azotobacter. They are present in association with roots of the tropical grass, right? So recently you can say that the strains of Azospirillum from the roots of grasses, rice, maize and sorghum, they are proved to be efficient nitrogen fixers when compared to the others. So coming to the next one, that is free living bacteria which act as biofertilizers. Let us see which are those. Okay. So there are certain free living soil bacteria. They are asymbiotic in nature. They are free living in the soil. Okay, so we can call them as the free living soil bacteria. They fix the atmospheric nitrogen actively even under aerobic conditions. So as I had told you, symbiotic nitrogen fixing bacteria, they work under anaerobic conditions, right? But here they can fix in the presence of oxygen itself. Okay, so one example you have Clostridium is an example, then you have Bacillus, also Azotobacter is an example. So the bacteria Azotobacter, when grown along with the other crops, it leads to higher yield and they reduce the nitrogen requirement also. Okay, coming to the next one, that is Cyanobacteria. So apart from bacteria, also certain blue-green algae or the cyanobacteria, they also act as nitrogen fixers. So some of the nitrogen fixing cyanobacteria can be seen in the rice fields such as nostoc, anabina. So here then also you have oscillatoria, another example. So here in them they have thick walled cells, okay, specialized cells called as the heterocysts, which are the site of nitrogen fixation, and they fix the nitrogen nearly for 30 kg per hectare. So these are being widely used as biofertilizers in different industries. And cyanobacteria like nostoc, they are also present in symbiotic association. They live in the coralloid roots of cycas plant. Okay, so nostoc can be found in symbiotic association also. So, and anabina, another example, it is also present in the leaves of azolla. So azolla, anabina, they are also in symbiotic association. Anabina occurs as an endophyte, right? So in the leaves of azolla. So the cyanobacteria here, it fixes the atmospheric nitrogen, and anabina, some species of anabina and also nostoc, as I told you, they live in the coralloid roots of cycas. And also this azolla and nostoc are added to rice fields to increase the rice yield by more than 50%. So this is how they are useful, okay, as biofertilizers. Another example we have Mycorrhizae. So Mycorrhizae, they are also called as the fungus roots. So they are also efficient biofertilizers. Myco means fungus and rhiza means roots. So commonly they are also called as the fungus roots. So it is a distinct morphological structure which develops as a result of association between the fungus and roots of certain specific higher plants. Okay, so these plants, they suffer from nutrient scarcity. Most of the time it is phosphorus and nitrogen deficiency might be there in those plants. So this association helps them to get the supply of the deficient nutrients and also other benefits like resistance to pathogens, root-borne pathogens, tolerance to salinity, drought, etc. So it also helps in the overall growth and development of the plant. So one example is mycorrhiza are formed by many members of the genus Glomus. So this is one good example. Then in detail, if you study about the functions of this mycorrhiza, as I told you, they help in the absorption of the nutrients, because these plants generally they suffer from nutrient scarcity, especially phosphorus and nitrogen. So when the fungus are present in association with the roots, they help to overcome this deficiency. And also resistance to many pathogens, tolerance to salinity and drought, and also it helps in the overall growth and development of the plants. So coming to the types of mycorrhiza. So depending upon the association, how it is present, the mycorrhiza are of two types, ectomycorrhiza and endomycorrhiza. So ectomycorrhiza here, they are found on the root surface as a mantle. So here the fungal mycelia, it forms the mantle, that is the outer sheath, okay, on the root surface. So from the mantle, the fungal hyphae, they penetrate into the root cortex and then in as well as into the soil. So examples here you have oak, eucalyptus, pines, okay. So here they absorb water and minerals and supply it to the plant. The other example, the next type, as I told you, it is endomycorrhiza. Here the fungus does not make a mantle on the root surface. Instead, it produces loosely interwoven hyphae. They are found inside the cortex of the roots. Example, the orchids. So these hyphae, they penetrate into the cortex of the root as well as into the soil, and then they absorb the phosphorus and supply it to the plant. So this is how these mycorrhiza work. Okay. So this is about the uses of microbes in as biofertilizers. So in this chapter, children, we have come across the different advantages or the benefits of microorganisms. So many a times we study about microorganisms only as pathogenic organisms which are infectious to the mankind or the other living organisms. But by studying this, we can say that yes, microbes are also beneficial to the mankind. There are different uses of microbes in the development of the mankind. Okay, so in this chapter, children, study the use of microbes in the different fields. Okay, so those are the important points what you have to remember here. So this is the end of the chapter. Thank you very much.